Hello again and welcome back to the metabolism series. This is the nitrogen metabolism and today we're going to focus on amino acid synthesis. So let's go. So here we are with the glycolysis. Okay, why glycolysis? Because from these metabolites and some of the Krebs cycle as well, we're going to be able to synthesize amino acids. So here we are. From glucose 6-phosphate, do you remember? We can get the ribose 5-phosphate. And the ribose 5-phosphate from the pentose phosphate pathway. The ribose 5-phosphate give us histidine. If we continue in the glycolysis, we have the free phosphoglycerate. Free phosphoglycerate, if it enters the pentose phosphate pathway, it will create erythrose 4-phosphate, which we're going to use in the next step. So free phosphoglycerate will give us serine, and this is the serine family. If we use the tetrahydrofolate, if you remember from the last video, we are going to move the carbons and we're going to get glycine. And if we use the S-adenosylmethionine, we're going to get the cysteine. This is the serine family. If we continue the glycolysis, we get the phosphenol pyruvate. Phosphenol pyruvate, together with the erythrose 4-phosphate, they're going to create the tryptophan, the phenylalanine, and the tyrosine. Here we have to mention that phenylalanine can be converted into tyrosine directly, as we saw in the first video. Then we have pyruvate, and pyruvate will give us alanine, as we saw in the alanine transaminase. And then leucine, isoleucine, and valine. These are branched chain amino acids. Then the pyruvate is going to be converted into acetyl-CoA, and then acetyl-CoA together with oxaloacetate will create the citrate. And this is the citrate cycle or Krebs cycle. Citrate is going to be converted into isocitrate and then to alpha ketoglutarate. And alpha ketoglutarate will give us glutamate, and glutamate has its own family. Glutamate will give us glutamine via the glutamine synthetase, and then arginine and proline. If we continue the Krebs cycle, we will get to oxaloacetate. And guess what? We have the conversion to aspartate. Oh, this is the aspartate aminotransferase. And aspartate has its own family. The asparagine, which we saw in the degradation, that asparagine gets converted to aspartate. And then lysine, methionine, and threonine. Pretty easy. So these are essential amino acids: the histidine, the tryptophan, the phenylalanine, the leucine, the isoleucine, the valine, the lysine, the methionine, and the threonine. They are essential. That means we cannot synthesize them really. So if we look at this diagram, we will have to cut these parts of the synthesis of the phenylalanine, tryptophan, all the essential amino acids. We will not be able to synthesize them. But plants and other microorganisms, for example, they can. So this is an integration of how they do it as well. Just to remember that we have to get them through the diet. Now, if we compare the synthesis to the degradation, there are some things they have in common. Okay, so starting from the phenylalanine to tyrosine, it happens here as well. So there, as you can see, the family is consistent. We have the aspartate family with the asparagine and the aspartate, of course. The tryptophan and the phenylalanine and tyrosine are here. The alanine is going to give us pyruvate, so this is exactly the reverse. And then the glutamate to alpha ketoglutarate, this is exactly the same. The glutamate family is the same of the degradation, except for the histidine. that some organisms synthesize it using ribose 5-phosphate, but they're very similar. Degradation and synthesis. Okay, so that was it for this video. The amino acid synthesis is done. I hope you liked the video. If you did, click the like button below. I will leave this diagram in the description so you can download it and study it very well. Subscribe to the channel if you're new, and I see you on the next one.